Welcome, everyone. Thanks for joining today's webinar on quantitative evaluations of immunogenicity assessment. I'm Jenny Yang, a Senior Director of Clinical Pharmacology at Atlas Pharma, and I will be your moderator for today. This session is presented by the Immunogenicity Working Group from the IQ Consortium, which conduct an industry-wide survey on current practices in immunogenicity risk assessment and data analysis. Today, we will focus on the data analysis part, with a second session on the immunogenicity risk assessment coming soon. So please stay tuned for that announcement. Here is a quick overview of the IQ Consortium. It's a non-profit organization founded in 2010, consisting of nearly 40 pharmaceutical and biotech companies. Its mission is to advance the science and technology to develop high-quality medicines for patients. And as you can see, it addresses many aspects of the drug development. Today, we will focus on the immunogenicity aspect. Biotherapy molecules become increasingly important for the pharma industry as well as in patients' lives, with a surge in approvals in the last decade. However, these therapies can trigger immune responses. Although our understanding of these responses has improved, challenges remains in identifying meaningful effects and communicating them in the product labels. The survey conducted by the working group aims to tackle these challenges and explore further opportunities. Our main presenter today is Dr. Carrie Kowalski, a director of clinical pharmacology at Pfizer in San Diego, where she works in both early and late-stage oncology development. Her work in deconvoluting the presence of ADA on clinical outcomes, as well as potential predictor for ADA incidence, has been informative for asset-level decision-making and regulatory interactions. She also helped to standardize Pfizer's immunogenicity reporting process and served as the primary contact across several areas, guiding team on the Immunogenicity Advisory Council. Here is our agenda for today. Carrie will present an overview of the survey result and IQ recommendations. After that, we will hear 4 case studies from the working group members, followed by a panel discussion for Q&A. Now, without further ado, I would like to hand it over to Carrie to begin the presentation.
Thank you, Jining. I'd now like to take a few moments to walk through what is immunogenicity and why we assess it. Administration of a biotherapeutic molecule can result in an immune response, which is often measured by anti-drug antibodies, or ADA. A variety of factors, such as characteristics of the drug product, in addition to patient-specific factors, can influence the propensity to developing ADA. The presence of ADA does not necessarily result in clinically meaningful consequences, but immunogenicity in this context is an unwanted effect, which has the potential to influence PK, safety, or efficacy. A subset of these ADA can be neutralizing antibodies, or NAB, which prevent binding of the biologic to the target of interest. Over the past 10 years, there have been a number of initiatives throughout industry and regulatory bodies to harmonize analysis and reporting of ADA data for biologics. A number of these focus on assay development and characterization. However, more recently, a focus on communication of clinical impact to prescribers has been a point of emphasis. There appears, however, to be variability in the methodology used to tease out the impact of immunogenicity to PK, safety, and efficacy outcomes. The IQ Working Group, with representatives from the companies shown in this slide, was formed to help elucidate methodology used across pharmaceutical companies to evaluate the impact of immunogenicity to clinical outcomes. The results of this effort were published in a February issue of CPT and are the focus of today's discussion. A survey was conducted over 20 member companies, with 17 companies participating. As analysis methodology can differ based on the characteristics of the molecule, the survey was distributed with the intent to capture analyses and resulting conclusions for individual molecules. This survey captured elements of molecule attributes, immunogenicity characteristics, analyses conducted and methods used, along with resulting conclusions and recommendations. As the focus of the survey was methods to assess clinical impact of immunogenicity, the survey was limited to molecules with clinical experience. Of the 93 molecules which were represented in the survey, approximately half reported being in the post-approval stage. Additionally, approximately half of the molecules were monoclonal antibodies, with only a small portion identified as cell therapies. As a result, the IQ was unable to draw conclusions for cellular immunogenicity, and the focus was impact of immunogenicity as measured by ADA. A wide range of immunogenic potentials were represented, with approximately half of molecules reporting less than 10% ADA incidence and 25% reporting an ADA incidence of greater than 50%. All modalities reported developing some degree of ADA. Although ADA incidence cannot be used to directly compare immunogenic potential across molecules due to differences such as assay sensitivity, this was an important data point to collect, as immunogenicity is a post hoc observation and the feasibility of analysis methodology may be dependent to a certain degree upon having a sufficient portion of the population with which to compare results. In addition to an ADA assay, ADA characterization and downstream analysis can be dependent upon the sampling rate within the clinical development program. A majority of molecules reported ADA sampling frequency occurring more often than every other month in registrational trials, and a majority reported ADA onset between 2 weeks and 2 months following the first administration of the molecule. A majority of molecules reported persistent ADA were detected. There can be various factors which influence the rate of ADA in the clinical setting, including population and dose dependencies. The most common analysis conducted to tease out these potential differences was single dose versus multiple dose, while the least common being a comparison of healthy volunteers versus patients. In most cases, the dose dependency of ADA incidence was either not evaluated or was not found to be dose dependent. When the rate of ADA was influenced by dose, directionality of this change varied, with 60% of molecules reporting decreases of ADA with increasing dose and 40% reporting the opposite trend. With respect to analyses performed to assess clinical implications of immunogenicity, the first endpoint which was evaluated was ADA impact to PK. The most common characteristic used to assess impact was ADA status, for example, positive or negative ADA. In most cases, no association of ADA status and changes in drug exposure were identified. When ADA did impact exposure, more compounds reported decreases in exposure rather than increases in exposure. When surveying the methodology used to evaluate ADA impact to PK, a variety of methods were used, including descriptive statistics, model-based methods, or a combination of these. When the ADA incidence rate was greater than 25%, a combination of methods was most often employed. ADA has been implicated in hypersensitivity reactions. A majority of molecules in late stages of development assess the impact of ADA to safety outcomes by ADA status. Approximately 1/3 of these drugs assessed impact by ADA titers, which was more frequent when ADA incidence was greater than 25%. This makes sense, as subsetting by titer threshold or quantile may require a sufficient sample size for interpretation. These analyses were most commonly performed via descriptive statistics. However, about 15% of molecules used model-based methods. When evaluating ADA impact to efficacy, both ADA and NAB were commonly evaluated. The analyses were most often performed through descriptive analyses, such as between and within subject comparisons. More complex statistical tests or advanced modeling was seldomly performed regardless of the ADA incidence rate, with less than 20 responses indicating performing more complex testing, which would aid in deconvoluting the impact of ADA from other factors, such as exposure changes or patient predictive or prognostic characteristics. When conducted, this was primarily assessed for drugs in later stages of development. An assessment of the implementation of clinical strategies to mitigate ADA impact was available for 84 compounds. The majority of compounds, 77%, did not implement clinical strategies to mitigate impact of ADA. When ADA mitigation strategies were employed, these were more common for compounds later in development. In most cases, this occurred when ADA was found to impact drug exposure. The most common mitigation strategy used was frequent monitoring. The next most frequent mitigation included increasing the dose of the study drug. Use of immune tolerance induction and immune suppressor co-administration were also mitigation strategies, which were pursued less frequently. Immunogenicity data, including analysis and impact to endpoints, are provided in support of a biologic application. According to the survey, most respondents reported health authority questions on assay performance. The greatest number of questions appeared to be related to clinical impact of immunogenicity. Across USPI reporting, there was variability in ADA content included. Most included the ADA incidence of the drug. Although a majority reported including NAB incidents in the label, the clinical impact of NABs was not discussed for over half of the drugs. With the new draft guidance for immunogenicity content and labeling issued by the FDA in February of 2022, content uniformity across drug products is expected to increase moving forward. Overall, the survey was expansive, including 93 molecules which had at least reached the clinical stage of development across multiple surveyed companies. A variety of biologic modalities were included with a wide range of ADA incidence and characteristics. In general, the data analysis methodology to assess ADA impact outcomes varied across assets. ADA positive or negative status was the most common characteristic used to evaluate ADA impact outcomes, with NAB analyses performed about half as frequently. For ADA impact to PK, a combination of model-based and statistical methods were often used. However, univariate approaches were more common for evaluating ADA impact to safety and efficacy outcomes. Overall, approximately 25% of molecules surveyed reported clinically meaningful impact of ADA on PK. 8% reported clinically meaningful impact to efficacy, and 21% reported clinically relevant impact to safety. Differences in analysis methodology will evolve over the course of development. As over half of molecules represented in the survey were in post-approval, the survey has larger representation of compounds at the late stage of development. The majority of molecules represented in the survey were monoclonal antibodies with few cell-based modalities. As a result, cellular immunogenicity considerations are largely out of scope. To maintain survey anonymity, indication, and year of entry into clinical trials was not collected, which prohibited data dissection of trends over time across therapeutic areas. Differences in assays and definitions of ADA terminology may prohibit adequate cross-molecule comparisons and were not elucidated in this survey. Many thoughtful discussions were had amongst the IQ members, and consensus on the following considerations were found helpful to convey. In general, ADA data quality can vary across molecules. Therefore, cross-molecule comparisons are often inappropriate. This includes considerations such as different assays in addition to sampling design. Immunogenicity risks should be continuously reevaluated throughout development to inform ADA monitoring. For example, more frequent sampling may occur in early development to inform optimal sampling for assessment of impact to outcomes within the phase 3 program and beyond. Additionally, considerations such as when ADA occurs relative to the outcome may be an important consideration. For example, transient ADA may not have as large of an impact to long-term outcomes as persistent ADA. Similarly, the impact to outcomes may be dependent upon the magnitude of ADA response, where an ADA response threshold may need to be reached in order to result in clinically relevant consequences. This can be elucidated through subsetting data during analysis. Finally, the methodology of analysis of immunogenicity impact to outcomes should be selected based on the stage of development, as well as compound-specific factors. There are scenarios where advanced methods may be needed to deconfound the analysis. However, when advanced methods are used, descriptive analyses should still be included within the integrated summary of immunogenicity. When assessing the ADA impact on PK, safety, and efficacy, there is a trade-off between the simplicity of the analysis method and the information which can be elucidated. For assessing the impact of ADA on PK, a simplistic view, such as box plots by ADA status, may be confounded by patient-specific factors which may influence both PK and ADA, or whether increased exposures may impact ADA incidence. A more informative view may include analysis of ADA as a covariate within the population PK model, which can account for the impact of other covariates on exposure. A mechanistic model may provide further insights yet into mechanism of ADA generation or impact to exposure. Similarly, ADA impact on safety and efficacy can be assessed via descriptive tables or figures, but multivariate exposure response analyses may provide additional insights through accounting for potential confounding factors within the exposure response model to adequately isolate impact of ADA on response. In some scenarios, advanced statistical adjustment may be needed. Ultimately, sample size, the magnitude of ADA's effect on PK and outcomes, the impact of confounders, and the presence of time-varying PK are all factors which should be considered to inform the most appropriate analysis approach. Today, we are highlighting what has been done and is currently being done with regard to assessing immunogenicity impact to outcomes. However, we acknowledge that there is more to be done in this field to impact decision-making. For example, immunogenicity assays are molecule-specific and often not commercially available. This begs the question of when a known clinically relevant impact exists, what can be done in practice to enable early detection and potential intervention? Similarly, analysis methodology can vary across industry. Most companies employ descriptive statistics in analyses of ADA impact on outcomes. However, mechanistic models may be more informative, or multivariate methodology may be needed to deconvolute the results. However, when employing these methodologies, the ability to detect a signal may be dependent upon incidence, duration, or response magnitude. The most common categorization used by companies in ADA analysis was ADA positivity status. However, in the future, this may transition to identification of a clinically relevant magnitude threshold or a duration of ADA response. As we approach these analyses, there may be new methods such as machine learning which can help optimize the identification of these thresholds or streamline these analyses. Now I am happy to transition to my favorite portion of the presentation. Select working group members will each present a case study to demonstrate how some of these methods were applied in their respective programs. First, Jocelyn Liu, a Senior Director of Clinical Pharmacology and Pharmacometrics from Johnson & Johnson, will walk us through descriptive analyses used to characterize immunogenicity. Then, Insa Winzenberg, Director of Pharmacometrics and Research Fellow from AbbVie, will discuss a unique model-based case study for evaluation of ADA impact to PK. Benjamin Wu, a Director and Distinguished Scientist from Genentech, will then go through an example of model-based evaluation of ADA impact to efficacy. Finally, Laura Hamoro, a Senior Director of Clinical Pharmacology and Pharmacometrics at BMS, will discuss application of a QSP model to predict immunogenicity and PK impact. Jocelyn, I will now hand it over to you.
Thank you, Carrie. I'll be giving some examples from published data on descriptive analyses used to characterize the impact of ADAs on outcomes from phase 3 trials of sub-Q and IV golimumab in psoriatic arthritis patients. To start, we typically determine whether a participant is considered treatment emergent ADA positive or negative through a time point or date, depending on the data cut. In such a summary, if relevant, one could start with evaluating whether there were participants positive for ADA at baseline and whether these were or were not treatment boosted. This includes looking at the baseline titer and then the titer after treatment at the subsequent visits to see if the titer increased at least 2 dilutions higher than baseline to be considered treatment boosted. And then these participants, along with those who were newly ADA positive after treatment, would be considered to be positive for treatment emergent ADA. Peak titers for those with TEADA can be listed along with their frequencies. Then, another analysis that can be done is to look at the incidence of neutralizing antibodies through a time or date cut, where one could take the participants positive for TEADA, see how many have samples to be further evaluated in the NAB assay, and then determine if these participants were positive or negative for NAB, with a percentage of these based on a denominator of those positive for TEADA. Lastly, to determine the overall incidence of participants positive for NAB for the study, we use the denominator of the immunogenicity analysis set. On the right side of the slide, we have an example of a phase 3 study in psoriatic arthritis where patients may or may not be on concomitant methotrexate, which is a DMARD. So this table shows how the subgroups can be split out. And for brevity, we have the peak titers presented in groups with their frequency, but usually we have these split out. Also for space, I have simply added the NAB incidences in the bottom rows. This slide displays the more clinically meaningful data, where we are now looking at the impact of immunogenicity on PK. Could also do this on PD if there's a good biomarker related to the mechanism of action, and then for efficacy and safety. Here I'm only showing examples of PK and efficacy plots. On the left-hand side, we have a plot of the median serum drug concentrations by treatment emergent ADA status. The blue with the hash lines are the TEADA negative participants, while the solid orange line are the TEADA positive participants. As TEADAs would be expected to accelerate the clearance of the drug, we can see the steeper concentration profiles and lower serum trough concentrations. Some variations of this type of plot could be looking at individual profiles as spaghetti plots and color coding the TEADA negative PK profiles and superimposing the TEADA positive PK profiles on top in a different color to characterize the impact of ADA on PK. On the right side, we have bar plots of clinical efficacy response as used in psoriatic arthritis, with the ACR response being the percent of participants being able to achieve or not achieve the ACR American College of Rheumatology 20% responder criteria as a lower bar efficacy response, and then the ACR 50 as a next higher efficacy response. Could also consider ACR 70, ACR 90, or remission. Blue bars are the TEA negatives, and the orange bars are the TEADA positive participants. What we can see from this graph is that while participants may be TEADA positive, there are a good proportion of participants that are able to achieve the ACR 20 response, with fewer able to achieve the ACR 50 response. On this slide, the ACR response and PK concentrations were superimposed on the same plot, but this example has the bar groupings broken out also by peak titer groups, showing that the impact of PK and the pattern in efficacy response has more impact at higher titers. So such analyses can be done for safety events such as injection site, infusion site, or infusion-related reactions, as well as for hypersensitivity, though these events may be more rare. Not shown here due to limited time, but more analyses on ADA and NAB could be performed, such as looking at when the onset of ADA occurred, as well as the transience of the ADA incidence over time. So now I'll turn it over to Insa for a model-based case study.
Thank you, Jocelyn. With that, I want to get to a case study of adalimumab, where we developed a data-driven population PK model with delayed ADA development. Adalimumab, or also known as Humira, is a monoclonal antibody and TNF-alpha inhibitor. In this particular project, we had immunogenicity data from an ELISA assay, and in addition to that, from an ECL-based titer assay. And we conducted a stepwise approach to fit the population PK model. And the different steps of this model development were, first of all, that we assessed the performance of the adalimumab immunogenicity assays in predicting low adalimumab trough concentrations. And based on this assessment, patients were stratified or classified into a group where the PK is not impacted by ADA and a group where the PK is impacted by ADA. And this classification was done by the most sensitive assay measurement, which was the titer assay. And after that, adalimumab PK dynamics were first of all modeled independently of ADA development based on the group where the PK is not impacted by ADA. And this is in the figure in the middle part, the green part, which is essentially a standard two-compartment population PK model, which was fitted on this data status. And then as a fourth step, the middle part of that diagram was fitted, which is the delayed ADA synthesis part and the complex formation together with adalimumab. This slide shows now the model validation and also an additional ROC assessment. Regarding model validation, please have a look at the left-hand side where we show visual predictive checks of the developed model, and they are split into three different categories. The left categories are subjects that have a negative titer assessment. The middle part are subjects with a positive titer but a negative ELISA assay, and on the right-hand side, subjects that are positive in both assays. And as you can see, they have also increasing impact of immunogenicity on their PK, and all three categories are rather well captured by the model, which you can see by the observations depicted with the dots and error bars in black and overlaid by the median predicted PK profile in purple and the 5 and 95% confidence intervals in blue. Next to that, we performed a receiver operating curve assessment based on the ELISA concentration assay, but also a signal-to-noise measurement and the titer measurement. And for that, we assessed the capabilities of each of that assay to identify the subjects that have an impact on PK from the ADA development. And we rephrased that in terms of subjects who had at least 30% of serum concentrations either above a threshold of 1 microgram per mil, or subjects who had at least 30% of serum concentrations less than LLOQ. And as you can see from the sensitivity versus specificity plots, so the titer measurement or the signal-to-noise measurement were the most sensitive ones. But if you look at the precision recall plots at the bottom, there the ELISA assay actually performs equally well, particularly if we look at this big dot, which shows the performance of the ELISA assay with a standard threshold of 20 nanogram per mil, which is really near the optimal point of that precision recall curve. And that concludes my presentation here. So if you want to look for additional details on that developed models, please look into the source that is given here. And yeah, thank you so much. And I would like to hand over to Benjamin for the next example. Thanks.
Thank you, Insa. In this next example, we'll look at how disease modeling in oncology can be used to evaluate impact of ADA on efficacy. What you see on the screen is an ADA framework designed to support this type of evaluation. So what is shown here is when a therapeutic protein is administered, the body is exposed to drug, which can trigger the development of ADA or neutralizing ADA. At the same time, drug exposure is essential to drive efficacy, which itself can be influenced by various patient characteristics and prognostic factors. Particularly relevant for cancer immunotherapy, time-varying pharmacokinetics are often observed, such that patient-specific factors can affect not only the efficacy, but it can also affect drug clearance. As this publication highlights, patient factors can also affect ADA development. This link between patient factor and immunogenicity is relatively novel. So as you can imagine, these interconnected correlations make it challenging to isolate the true impact of ADA on efficacy, as many of these relationships can introduce confounding effect. Here are some data that illustrate the confounding effects that we just discussed. On the left, we see a figure from publications on NEVIL that highlights how differences in clinical response can lead to changes in drug clearance. Patients who experience disease progressions and essentially become sicker tend to show increasing clearance over time. This pattern has been consistently observed across multiple tumor types, including non-small cell lung cancer, renal cell, and melanoma. Now, when we look at the overall survival, it is well established that prognostic factors such as C-reactive protein can significantly influence the overall survival outcome. What's particularly interesting is the observation on the bottom right for atezolizumab. Here we found that patients with higher baseline C-reactive protein, again sicker patients, are also more likely to develop ADA. And this trend holds true across several cancer types. So the key questions become, how can we accurately assess the independent effect of ADA when the same prognostic factor influence PK, efficacy, and ADA development? The prognostic factor affecting each factor partially overlaps, but they're not completely the same, creating a complex web of confounding effect that we need to carefully evaluate. To begin addressing this challenge, we explore several approaches, one of which involves using a tumor growth inhibition overall survival or TGIOS model. This model includes both drug-specific and disease-specific components, as illustrated in the diagram. It integrates baseline patient characteristics along with on-treatment tumor dynamics to predict the overall survival across multiple cancer types. One of the key challenges we face is that ADA is a post-randomization variable. That means the baseline characteristics of ADA positive and ADA negative patients may no longer be balanced, leading to a potential confounding when interpreting ADA impact. The strength of the TGIOS model lies in the ability to simulate balanced distributions of baseline prognostic factors. This allows us to isolate better the independent effect of ADA on overall survival. What we have found is that if you don't account for imbalances in baseline factors, The ADA positive subgroup often appears to have worse hazard ratios. However, as shown in the figure on the right, once you adjust for those imbalances using TGIS model, the ADA positive group performed comparably to the ADA negative group. This suggests that the initial observed difference may be driven more by prognostic imbalances rather than by ADA itself. It is worth noting that TGI-OS model is just one of several advanced methods to address the confounding effect of ADA, and when we apply other approaches, it also led to similar conclusions. With that, I'll turn over to Laura Hamuro from BMS, who will present the final example using a quantitative system pharmacology model.
Thank you, Ben. Let's transition now to the mechanism of immunogenicity. The advantages of having a mechanistic model to predict ADA and PK impact is to allow for extrapolation and prediction beyond the currently tested conditions, and specifically to test hypotheses related to the mechanism. On the left side of the slide, the innate and the adaptive immune responses are illustrated. Examples of key factors contributing to increases in biotherapeutic immunogenicity are highlighted red, and decreases in immunogenicity in green. The QSP model includes system parameters inherent to the biology and compound parameters specific to the drug. Key system parameters capture affinity maturation, memory responses, timescale of T and B cell activations, and HLA frequencies. Key compound parameters include antigenicity predictions, PK, molecular weight, dosing regimen, ADA assay sensitivity, fraction of antigen-specific naive T and B cells, and antigen dendritic cell uptake rates. The top right illustrates how key compound parameters and system parameters come together. The model parameters representing the biology are calibrated against the prospectively compound-specific inputs indicated on top. Once model predictability is confirmed prospectively with a new clinical data set, ADA predictions for what-if scenarios can be conducted. For example, what if immunosuppressive conmeds are administered together with the drug? What if the population changes? What if the dose level and dosing interval changes? What impact does that have on ADA? Can be addressed with the QSP model. Here's an example of an immunogenicity QSP model that we published a few years ago. We had an asset in phase 1 being tested in healthy adults with a known immunogenicity incidence, and we wanted to predict how ADA might change in a disease population in which they were undergoing combination therapy with an immunosuppressive standard of care. We started with an off-the-shelf QSP model that was developed and published by Chen, Hinkling, and Vicini. This is often referred to as the CHV model. The model successfully predicted single-dose ADA, but over-predicted multi-dose ADA. That data is not shown here, but it is detailed in the paper. We reasoned that if we included a Treg compartment to capture immune tolerance and immune system downregulation, we could then predict both single and multi-dose ADA. In the PK/PD submodel, we made assumptions of one-to-one binding of ADA to drug to accelerate clearance. And in the middle stacked plots, we demonstrated no ADA impact on PK. In the legend, orange is ADA negative and blue is ADA positive, which are overlapping and superimposable to the pink color to indicate no change in PK on the top or PD on the bottom. We then used the model to predict ADA incidence with increasing concentrations of corticosteroids. On the far right plots, the model is predicting a reduction in ADA with increasing corticosteroids at increasing dose levels, with red being ADA prediction over time without the corticosteroids. You know, QSP models of immunogenicity are increasingly being used, and the future of these models is indeed promising and will continue to evolve, ultimately to improve drug development and patient safety.
Thanks, Laura. And that concludes our presentation part. Thanks to all the presenters. Before we begin the panel discussion, I would like to acknowledge the working group members. A special thanks to our co-chairs Benjamin Wu and Susan Richard, along with all the contributors for their hard work and commitment to this initiative. We also greatly appreciate the support from the IQ Consortium in helping to develop this presentation. Now we can open the floor for the Q&A. We invite all the panelists to join us live. And audience, please feel free to submit any question you have through the chat. Thank you.